Symposium. It's really my great pleasure to welcome Sheng Li as our speaker today. And Sheng did her both her graduate work and then move up to become an instructor at Weill Cornell in New York City, working finally with Ari Melnick, who continues as one of her collaborators. And then in 2016, she started her own group at the Jackson Laboratory for Genomic Medicine, which is in Framingham, Connecticut, where she's risen through the ranks to become an associate professor. And she also has an adjunct faculty appointment with the University of Connecticut School of Medicine, which is on the same campus. And I think most importantly, Sheng and her lab have just made enormous contributions to some of the computational approaches to analyze large data sets, single cell data sets, methylation and hydroxymethylation, genome-wide data sets. And it's really enabled a lot of fantastic research into understanding heterogeneity within cancer, such as heterogeneity within AMLs, heterogeneity that's caused by aging. How do our cells become sort of drift away from that sort of evolved norm as we get older? And what are the implications for that for functionality and for potential adaptation to change that could lead to oncogenesis? So these are really some of the fundamental questions that her lab has been approaching. And we've been collaborating with them as well. She's incredibly collaborative, which is, I think, a feature not only of a good scientist, but of someone who's going to be successful. So without further ado, we'll let Sheng take over and give her talk. Oh, and the title of her talk is Deciphering Cellular Epigenome Heterogeneity and Evolution in Cancer Within an Aged Ecosystem. And I almost forgot, and Adela would kill me. So we'd like to present you with, this is our Cancer Center Award. It's the Cancer Warrior Award, and it's for your contributions to cancer research.
Oh,
so
thank you. Really appreciate that.
Sure. And I have a box to put it away in later.
Thank you so much. Yeah, I didn't expect that. I really feel honored to be invited here and then share our ongoing work and also some of the work we've done in the past in the space of like cancer, data science, and aging. And thank you for very nice introduction by James. And it's been really like a fantastic experience in this collaboration with James and learn a lot, especially thinking about how to position ourselves in the interface of aging, cancer, and data sciences. So today I'm going to share with you guys on an ongoing effort to understand leukemic onset during aging, and also especially from the perspective of cellular epigenome heterogeneity and evolution in the context of aging. So I would like to start with my vision of cancer research. So there's as we've been like really looking into like a cancer systematically for over a decade ago, over decades. So there's a lot of emerging topics that really deserve more attention and really like these are more collaborative work, especially in the topic of aging, inflammation, and senescence. On the other hand, what I've been observing is that technology really helps us to transform our understanding of cancer and biology, many things in general. And what I see like emerging technology that really are transforming the landscape for cancer research are single cell omics, long-read omics, as well as spatial omics. And an important factor to connecting the two parts, like the major topics in cancer research and also technology or data science. And that's what we do in the lab every day. And the goal eventually is to translate the rich information from the advanced omics technologies that we can eventually contribute to the patient care. So that's the long-term goal here. And specifically, the question that I ask and the focus of my lab now is to understand how somatic mutations alter the epigenome of blood cancer and gene regulation in the context of aging, so that we can use via like algorithm development and also integrative mining of high throughput data, including like those omics data type that I mentioned earlier, so that we can lay the foundation for crafting future epigenetic therapeutics and preventative strategies to expand health span and lifespan. Where I see that there's a lot of like ongoing like initiatives on this campus that really resonate a lot on what we are interested in like in the daily lab work. So cancer has been like well recognized as the disease of aging, and thanks to a lot of pioneer work that James have done like over a decade ago, many decades ago. And a lot of like what we've been like really looking at from the like SEER database to looking at the age with the incidence levels of different cancer, right? As shown in like one of the many papers that James has done in the past. Like cancer happens many after like 90% of cancer happens at the age of 50, and we don't really know like why. And there's just start like there's growing research start to looking into this angle, adding the aging dimension to the cancer research, and this is partly because the aging population has been expanding like rapidly, and it has been estimated that by the year of 2060 there will be more than 23% of the population that have like age over 65. And interestingly, many of the biological processes that underlie the hallmarks of aging and hallmarks of cancer overlaps, including the epigenetic dysregulation, genomic instability, heightened inflammation, and increase in cellular senescence, and thus point to the existence of biological connections between the two. On the other hand, most of the preclinical studies use young mouse, which is equivalent to the older adolescent in human. And also in clinical trials, elderly, especially those over age 75, are underrepresented. Although the trends are changing, like thanks to like the FDA issued guidelines to include more elderly patients, the study of aging in cancer is still very much at its infancy. So I've been working on the cancer epigenomics for over a decade, and that hits on both the hallmarks of aging and cancer. And epigenetics is the study of gene regulation without changing the DNA sequence. And this kind of changes is heritable, at the same time reversible, as being shown in many of the studies that coming out of this campus here. So epigenetics, the study of how cells like control gene activities without changing DNA sequences, right? And like our genes plays an important role in our health, and this, but also to our behaviors and environment, such as what we eat and also how physically active we are. And epigenetics is the study of how our behaviors and environment can cause heritable changes that affect the way our gene works and basically just turns on and off. And the epigenome is the collection of all the epigenetic marks on the DNA and the protein wrapped on the DNA, including those on the DNA, the DNA methylation, 5-MC, and hydroxymethylation, 5-HMC, as well as on the histone, the methylation, acetylation, and anything that are non-genetic, including the chromatin interactions from the 3D genomics data, Hi-C, and non-coding RNAs. And our epigenetic changes as we age, both as part of the normal development and aging, and in response to our behavior and environment. Notably, more than 50% of the human cancer harbors mutations in genes that encode regulators of the epigenome. These regulators including writers that establish epigenetic marks, such as DNA methyltransferase, histone acetyltransferase, and also erasers that remove the epigenetic marks, including the TET family of methylcytosine oxidase and histone deacetylase. And the plasticity of the epigenome owes much to this existence of these erasers and in cooperation with the writers. And tumor cells routinely use the epigenetic process to ensure that escape from chemotherapy and also the host immune surveillance. And epigenetic reprogramming can give rise to essentially each classic hallmark of cancer. And for example, by tumor suppressor silencing, oncogene activation, or cell fate transitions. That's why there's a growing emphasis on recent drug development. It has been targeting the epigenome, including the DNA methylation and histone modifications. And several of them has already been approved by the FDA. And currently there are like at least nine FDA approved epigenetic agents, including the 2 DNMT inhibitors such as 5-azacitidine, and also isocianate dehydrogenase IDH1/2 mutant inhibitor. These are target therapy, ivosidenib and inacitinib, and others targeting the histone modifications. And my lab have been using the hematopoietic system extensively as a model for us to understand the demethylation regulation and epigenetic regulations. And hematopoietic stem cells can self-renew and differentiate into all kinds of blood cell types, including immune cells, a process called hematopoiesis. And many epigenetic regulators are the gatekeeper of hematopoiesis, including those genes that regulate demethylation, enzymes that have opposite function of demethylation, such as TET2 that can remove the DNA 5-MC from the DNA, and also DNMT3A that can add the methylation to the DNA, and IDH1/2 that regulate the TET2 activities. And we have shown that TET2 loss leads to the gain of cytosine methylation, the 5-MC, and also the 5-HMC loss and histone acetylation loss, and gene silencing that eventually blocks the differentiation and drives the hematopoietic malignancies. And ML is predominantly a fetal hematopoietic malignancies. It occurs when the block of the myeloid differentiation happens, and also increase in the malignant self-renewal. And the median age for ML is 68. So that's right in the realm of like majority of the cancer happens at the like a much older age. And it's an aggressive cancer that starts from hematopoietic stem and progenitor cells. And the 5-year survival rate is 26%, and the relapse rate, depending on the subtypes, is on right around 50%. And previously, the large-scale sequencing study of the ML patients has discovered that the recurrent mutations, many in the gene encoding the epigenetic regulators, such as the DNMT3A, the 5-MC writers, and also like TET2 and IDH1/2, those 5-MC erasers or TET2 regulators. And these mutations also start to emerge as blood cells as we age, a term called clonal hematopoiesis. And just what we are like enjoying the AACR annual meeting this year in April, there's like a series of papers published on Nature, starting from rooted from the TraceX consortium that highlight the importance of studying the cancer evolution in cancer study, as it has been discussed many years later. Nothing in cancer biology makes sense except in the light of evolution. Really underline the importance of looking into the evolution of cancer cells. And this is not just in the solid tumor, but also like even trace back to like over a decade ago, whole genome sequencing study, or more recently, the single cell mutation analysis has been highlighting the clonal evolution in the myeloid malignancies in AMLs. And so most of the study in the cancer evolution has been primarily focused on the genetic changes and the genetic clonal evolution. On the other hand, epigenetic marks has great plasticity and may vary over time or through the exposure to the environmental like stimuli. Thus, there is a potential for epigenetic diversification to emerge in tumor cells populations and to change during the disease progression. And such kind of heterogeneity may provide options for the subset of cells to manifest selective growth and survival advantages to the escape immunological surveillance and also therapy beyond genetic heterogeneity, especially for cancer with low mutation burdens. So that's why we have been really looking into the epigenetic plasticity and also the heterogeneity that especially in the like cancer cells. And that's the overall hypothesis that this kind of epigenetic heterogeneity can allow the malignant cells to sample alternative transcriptional programs and adapt to their environment. So ML is a system that well suited to examine and also study the question about epigenetic heterogeneity, as this kind of like tumor generally manifests a relatively positive or like of genetic lesions, but still showing genetic clonal complexity and clonal evolution during the progression. And previously used the meth clone that I developed to analyze the single molecule bisulfate sequencing data. We previously showed for the first time that hematopoietic clones from relapsed ML patients manifest selection of malignant cells harboring specific epigenetic alleles. And this kind of manifestation is linked to the disease severity in patients in ML, and specifically their time to relapse. At the same time, this is not just ML, right? Over years, among ours and others' work, the evolution and heterogeneity measured by single molecule demethylation sequencing has shown that this kind of epigenetic heterogeneity and evolution is widely manifested in both hematopoietic and the solid tumors, including the chronic lymphocytic leukemia CLL and also the B-cell lymphoma DLBCL, and glioma, lung adenocarcinoma, breast cancer, and Ewing sarcoma. So this is a widespread phenomenon in terms of the somatic evolution and epigenetic heterogeneities in cancer. In addition, it has been shown that the clinical relevance of the cancer somatic evolution and epigenetic heterogeneity across the different hematopoietic and solid tumors. For example, in the Ewing sarcoma, the epigenetic heterogeneity has been associated with the tissue of origin and metastasis. And in CLL, the epigenetic heterogeneity level, the higher the epigenetic heterogeneity levels, the worse the overall survival of the patients. And in terms of relapse, it's not just ML, but DLBCL also has been manifest the association between the higher epigenetic heterogeneity and also the relapse. And in glioma, the time to surgery. That highlights the importance of looking into the epigenetic heterogeneity and cancer evolution. But if one's interested in the epigenetic heterogeneity, how you can quantify that? So one of the platforms that we have been looking extensively since over a decade ago is the bisulfite sequencing data. The reason is because these can provide the single base, single molecule phase demethylation patterns for you to infer the cellular epigenetic heterogeneity across population of cancer cells. And the principle is straightforward. If a cytosine is methylated, then after bisulfite conversion, it will be protected. Thus, it will remain as cytosine in the Illumina sequencing output. At the same time, if a cytosine is unmethylated, then it will be converted into uracil by bisulfite conversion, and eventually in the Illumina readout, it will be thymidine. So by just calculating the number of reads supported cytosine divided by the total number of reads from cytosine and thymidine, you can assessing the single base demethylation patterns. And in general, whole genome bisulfite sequencing can give you about 28 million CpG sites. And then reduced by representative bisulfite sequencing can give you about 46 million CpG sites covered. And demethylation capture sequencing is another way. Depending on the probe design, you can also get similar range of CpG sites. So my lab have been, among many others, has been utilizing the epi alleles to trace clone evolution and infer epigenome heterogeneity. And there are several methods to assess the epigenetic allele diversity at single molecule resolution, from the epigenome evolution to epigenetic heterogeneity. So epi alleles, although it's not identical in the concept of genetic clones, it helps provide a basis to measure the population diversity among individual cancer cells. And it's essentially serves as a native barcode to trace the cancer evolution. So in our previous work, we developed a computational tool called Methlon to use the single molecule demethylation bisulfite sequencing read to measure the epigenetic allele repertoire shift over time. For example, from normal to diagnosis or from diagnosis to relapse in ML. And it's essentially just to trace what's the compositional change of the DNA methylation patterns, which from a set of 4 CpG sites that harbors compositional change from of epigenetic alleles from different time points. And the epigenetic allele repertoire shift serve as essentially here a molecular clock for cancer evolution measurement. And this is available as a C++ program that I developed, published in 2014. And in addition, if you're interested in the epigenetic heterogeneity, there's multiple options that you can assess them. One is the epigenome instabilities, and that assess the locally disordered demethylation. Initially published from the Cathy Wu lab by Dan Landau and now he opens lab in the Weill Cornell Medicine. So that was initially published also in Cancer Cell in 2014. And essentially it's measuring what's the percentage of the DNA fragments that represent the discordant reads that represent the different demethylation patterns out of all the reads measured from the same locus. And those concordant reads are those that only have unmethylation status or fully methylation status. So now we also implement this measurement, epigenome instability or epimutation rate in our R package, EpiHit, as our bioconductor R package. And in addition, if you're interested in the cell-to-cell variations, another option is called epipolymorphism that assesses the cell-to-cell variations, and is a concept that borrowed from the population genetics, and essentially it's asking if you are sampling 2 DNA fragments from a population of cancer cells, what's the probability of sampling 2 different DNA fragments that manifest different DNA methylation patterns? And that's also now this measurement of epipolymorphism is incorporated in the EpiHit R package. And the higher the epipolymorphism, then the higher the cellular cell-to-cell variations. And as an overview, using the single base, single molecule phase demethylation patterns, one can start to quantify the epigenome in heterogeneity and evolution. And just imagine if you group all the DNA fragments you sampled from the a population of cancer cells, and each row is a DNA fragment, and each column is a CpG site. Then if you're looking at a one time point, then you can start to assess the cell-to-cell variation across the DNA fragments, and that's called epipolymorphism. And then you can also assess the epigenomic instabilities for each DNA fragment across the CpG site at single molecule resolution. And then if you're interested at the different time point, how does the cancer cell population evolve, then you can use methclone to measure the epigenome evolution, essentially asking what's the epi-allele repertoire shift over time, the compositional change of DNA methylation patterns from one point to the others. And my lab have been developing like this EpiHit and also methclone to assess the epigenome heterogeneity and the evolution, not just for the ML, but also broadly for any type of like bisulfite sequencing data that are made available, and preferred with reduced representation bisulfite sequencing, just because they have much higher coverage for those regions that of medium and high CpG density. And in general, bisulfite sequencing have an advantage of like higher coverage across the epigenome. Like in general, you can get like 4 to 6 million CpG sites, and also higher convergence of like cytosines across DNA fragments, replicates, and groups compared to the single cell molecule, single cell bisulfite sequencing data. And next, I'm going to talk about the application of our package to studying the ML heterogeneity, because this is like a cancer of like lower genetic mutation burden and higher epigenetic alteration. So specifically, previously, we found that the ML patients with higher epigenetic heterogeneity exhibit poor event-free survival for those ML patients that sampled at diagnosis stage. And this is a cohort of 119 patients that have the bisulfite sequencing data we get from the Munich lab at Weill Cornell Medicine. And this suggests that epigenetic heterogeneity can provide malignant HPCs, hematopoietic stem and progenitor cells, with an additional layer of fitness beyond genetic heterogeneity. However, the source of epigenetic heterogeneity and its potential link to the cancer-associated mutations remains unknown. And understanding whether and how mutations, especially those in the epigenetic regulators, can destabilize the epigenome could provide the important insights into population fitness and also the resilience and relapsing nature of this cancer. So here, in collaboration with Ari, we start to like ask the question about what's the source of epigenetic heterogeneity, and also importantly, given those epigenetic therapy available, whether this epigenetic heterogeneity we observe like also manifests in the ML, whether it's reversible or not. So you can think of 2 scenarios here. First, it's possible that epigenetic heterogeneity is purely stochastic phenomenon that is unrelated to somatic mutations and is a byproduct of ML disease progression. Second, it's also possible that epigenetic heterogeneity is induced by particular somatic mutations, which implies that such mutation might in some way disrupt the regulatory states that could normally be strictly controlled by the epigenetic regulation. And in this latter case, epigenetic heterogeneity might be predicted to proceed and perhaps even contribute to the malignant transformation. So here, in order to answer these questions, we explored the epigenetic profiles of a cohort of clinically and genetically annotated ML patients to investigate whether any of the canonical somatic mutations occurring in ML might be linked to the development of epigenetic heterogeneity. And these clinical specimens was processed by bisulfite sequencing and then targeted sequencing by the Melnick lab. And Xiaowen in the lab compared and contrasted the epigenetic heterogeneity in patients according to their genetic status. And she observed that specific mutations are linked to the defined level of epigenetic heterogeneity using the EpiHit package that she developed. And we know that TET2 and also like IDH1/2 mutations disrupt the cytosine methylation patterns. In particular, those like IDH enzymatic output is a metabolite that can activate the TET2 activity, while the mutant IDH can output an oncometabolites that inhibit the TET2 activities. Because the TET2 and IDH are epigenetic regulators for the DNA demethylation, then we decide to look into these two subtypes in the downstream analysis. And specifically, we asked whether epigenetic heterogeneity is caused by these leukemia mutations or just a byproduct of the leukemic transformation. So Alan Sheng from Ross Levine lab at MSKCC previously showed that the TET2 and the FLT3 mutations cooperatively induce ML. And FLT3 mutations are found in peripheral blood samples of 30% of ML patients, and one of the like a recurrent mutation that frequently found in ML, and it's commonly co-occur with TET2 mutations. So Xiaowen examined the LSK cells, so that's an early form of hematopoietic stem cells from TET2 knockout or mutant IDH2 knock-in mice, alone or crossed with FLT3 ITD mice. And the RBS and RNA-seq data were generated from the Levine lab. And by examining the LSK cells from these various mice before they're becoming transformed, they're still healthy, non-leukemic mice. Xiaowen showed that in the box plot, that TET2 in combination with FLT3 induce a higher level of epigenetic heterogeneity compared to the wild type and also single mutant mouse models. Similarly, IDH2 in combination with FLT3 induce also higher level of epigenetic heterogeneity. So the epigenetic heterogeneity here, because we are also looking at the healthy mice here. So it suggests that epigenetic heterogeneity is induced by the mutations and enhanced by the cooperation between somatic mutations, even before the overt malignant transformation. In addition, previously, we also showed that ML epigenetic heterogeneity with gene promoters is linked to the hypervariable transcriptional regulation. And that was using the single cell full length RNA-seq by fluid and C1 system. And that's over 6-7 years ago. So that's the time when we still only have a few hundreds of cells in total, completely different from the landscape now using 10x. But it's we still were able to like investigate the transcriptional heterogeneity at a single cell resolution and confirm this association between the epigenetic heterogeneity at the promoters and also the cell-to-cell variations. And in order to determine the association between the transcription variation and the downstream of leukemia mutations, Xiaowen compared the gene expression heterogeneity among the wild type IDH and the double mutant mice. And she found that the genes containing epi alleles with increased heterogeneity in the transcriptome levels compared to the wild type and single mutant mice. And also to understand the functional relevance of the epigenetic heterogeneity, she also performed the enrichment analysis using transcription factor binding site for those with increased heterogeneity in ML patients, and also like in the mouse models of matching like a genotype. And she found that the epigenetic heterogeneity arising in human and mice may affect similar transcription factors relevant to ML biology, including the HSC proliferation, self-renewal, and the leukemogenesis. So altogether, this data suggests that the epigenetic diversity can occur even prior to transformation as a consequence of somatic mutations, and perhaps even enable the cells to assemble in more transcriptome programs. However, the remaining question is that can epigenetic therapy reverse such kind of heterogeneity at both the epigenome level and also the transcriptome levels? So specifically, given the significance of the population fitness for disease outcome, and it's also intriguing to consider the concept of clonal reduction as a novel therapeutic target for ML. And there's no obvious way to reduce the disease clonality from a genetic perspective, right? But it's compelling to hypothesize that if DNMTI, such as azacitidine or IDH2 inhibitor inacitinib as an epigenetic therapy could mediate such effects at the cytosine methylation levels. By causing a reduction in the cytosine methylation across the genome, it would stand to reason that epi alleles could also become reduced in complexity. So here, we're also looking at the drugs that, like, for example, being approved by MDS and also like ML. And MDS, as you know, like is a form that ML can be developed from. It's either can be developed from the patients with MDS or just de novo developing from the healthy person. So Alan from Ross Levine's lab previously showed that azacitidine and inacitinib treatment induce an attenuation of the aberrant DNA methylation patterns and the transcriptional output, and result in a reduction in the leukemic blasts consistent with their anti-leukemic activities. So here we are specifically asking if these kind of drugs can reverse the heterogeneity we observed earlier. So here, we're looking into the bisulfite sequencing data and also RNA-seq data of the ML mouse models treated with the epigenetic therapy and also FLT3 inhibitors. And indeed, we observed that there is a significant reduction of the epi-allele diversity in the TET2 FLT3 double mutant ML models treated with 5-azacitidine, suggesting that a benefit of this class of drugs in ML could link to the reduction of population fitness. And this idea is further supported by our finding that the azacitidine also reduced the transcriptional hyperactivity or heterogeneities in ML mouse models. And perhaps this effect might also help to explain the potential benefits of priming the patients for chemotherapy by first exposing them to a DNMTI, and thus minimize the option of the tumor cells has to adapt to chemotherapy exposure, and also reducing the chance of particular cancer cells could escape the therapy. And to assess the relevance of the drug effect in human, so we explored the literature at that time, like that's what's available is the DM-methanol of 6 MDS patients treated with 5-azacitidine. So Xiaowen looked into the DM-methylation heterogeneity from these patients, and she find that there is a reduction of DM-methylation heterogeneity at day 29 of post-treatment. And this is true for 3 out of the 6 MDS patients. Next, we hypothesized that IDH2 mutant inhibitors inacitinib could also mediate such effect as the cytosine methylation levels by causing a reduction in the DM methylation across the genome. And notably, we observe a similar effect on the epigenetic and also transcriptional heterogeneity levels in the case of mutant IDH2 inhibitors. And thus, there's a similar beneficial effect on the epigenetic heterogeneity that may be also achieved by a specific reversal of the effects of epigenetic driver lesions, as opposed to the more global and non-specific action of DNMTI. And this suggests that a combination of IDH inhibitors and also chemotherapy holds promise as an effective approach for ML. And on a relevant note, like later, we also show that also find that there's a report from ANGM and then back into the Nature Medicine paper from the Jordan lab here in this campus, that the combination therapy of 5-azacitidine and the venetoclax together can help reduce the benefit the patient's outcome, especially increase by 5 months of elderly with ML patients. And this suggesting that the epigenetic drug therapy combination with a standard therapy may be an important clinical tools. And the single monotherapy venetoclax alone only represents modest activities, and similarly, just the azacitidine by itself. And in addition, in a more recent study on the ANGM, it's last year, It's also shown that ivosidenib, the IDH mutant inhibitor plus azacitidine, can increase the patient's median overall survival in the IDH mutant ML patient by 16 months. That really adds the excitement of like epigenetic drugs as serving as clinical tools to combine with other standard therapy to further enhance or improve the patient's clinical outcome. And thus FDA approved this combo therapy to treat newly diagnosed ML recently. So as a summary here for the first part, we show that somatic mutations drive specific but reversible epigenetic heterogeneity states in ML. And specifically, the epigenetic evolution and heterogeneity can predict the inferior clinical outcome of the patients. We develop also machine learning models that are composed of 28 loci that are able to distinguish the low risk and high risk ML patients. And somatic mutations are the driving force for the epigenetic heterogeneity. Epigenetic and also the transcriptional heterogeneity is reversible pharmaceutically, especially by the epigenetic drugs that either hypomethylating agent that are more global, or like inacitinib, the more targeted therapy, and suggesting that clonal reduction may serve as a novel therapeutic target for ML. And these findings also support rationale for future studies to rigorously trace epigenetic heterogeneity over time in patients with ML at relevant time points and to determine whether epi-allele reduction is associated with improved therapeutic response. So now that we know that somatic mutations can cause epigenetic heterogeneities, what are the role of aging and its causal role in epigenetic heterogeneity in leukemia initiation is still unknown. And this is an ongoing story that we are developing in collaboration with James and also Eric. So during aging, hematopoietic stem and progenitor cells capacity and immune functions declines with age. On the other hand, the bone marrow environment is associated with increased pro-inflammatory cytokines, and there are also like mutations that accumulate linearly with age, and DNA and chromatin modifications drift with age. And another age-related blood phenotype is cloning hematopoiesis. That happens when some hematopoietic stem cells make cells with the same genetic mutations. And these blood cells have different genetic patterns than the rest of your blood cells. And interestingly, these somatic mutations are also enriched in the genes that are frequently associated with leukemia. And because HSC, hematopoietic stem cell, can also produce immune cells, these cells can carry these mutations and eventually contribute to the dysregulated immune system. And it has been shown that the hematopoietic clonal hematopoiesis can cause hematopoietic malignancies such as ML, and also it can cause the cardiovascular disease. And there are emerging studies that shows that clonal hematopoiesis carriers is associated with increased type 2 diabetes and also the heart failure mortality and pulmonary disease. And now it's just started to show that in the recent Med archive paper that the CH carriers, clonal hematopoiesis carriers, is also associated with the Alzheimer's disease, underlying the importance, the clinical importance of studying clonal hematopoiesis. So you guys probably are very familiar with this result. Like previously, James has been shown like that the specific oncogenic events are selected within the age hematopoietic contents depend on the inflammation. When they transplant the young HSC cells carry the somatic mutations, oncogenic genes, and then transplanted into young old mice or old mice treated with the inflammation blocking drugs, IL-37. And interestingly, those oncogenic events only expand in the old microenvironment. And when they treated with IL-37 to abolish the inflammation, and this kind of oncogenic expansion doesn't exist anymore. So in this collaboration with James and Eric and also Hideyuki from UConn Health, now we're trying to understand what's the corporate impact of somatic mutation in tattoo, and also the age, the environment drives the epigenetic heterogeneity and eventually the leukemia initiation in the context of aging. And in particular, we are interested in like whether tattoo mutations in the aged mice can gain a higher heterogeneity than the young mice, and also what's the functional impact of such heterogeneities, especially when it's come to in the environment of heightened inflammation and also dysregulated immune HSC functions, how they contribute to like drive these cells to be more adapted to the age context as a clonal hematopoiesis, and also permissive to the transformation with the FLT3 ITD with age. And building on our prior work and also inspired by the evolution biologists. So here we propose that the natural selection has led to the tissue landscape that disfavor the phenotype altering mutation or epimutations through use. And the mutations or epigenetic changes that alter the phenotypes will tend to reduce the fitness of the stem cell or progenitors and promoting their elimination. Thus, progression towards the malignant genotypes, epigenotypes will require traversing a low fitness value, which is difficult. But in tissue damaged by aging and other insult, stem and progenitor cells will no longer be well adapted to this altered microenvironment and leading to the selection of new phenotypes that are more adaptive, including more malignant phenotypes that drives up the epigenetic heterogeneity in the inflammatory environment that eventually may promote clonal hematopoiesis and also leukemogenesis. So this is as part of the onco-aging consortium that we are studying the impact of aging on the cancer initiation. And specifically, we're combining the single cell sequencing and genetic barcode and aged host to perform the in vivo parallel tracing of clonal history and identity. So previously, as a proof of principle, my lab has generating the preliminary data from the same pool of hematopoietic stem and progenitor cells of the single mouse from 10x Genomics. And here, we met looking into the 3 age groups. And Ahmed, the postdoc in the lab, analyzed the 3 ages of mice and annotated these HSPCs, hematopoietic and stem progenitor cells. And he identified that long-term HSC aging signatures and found that the known aging markers, such as CD63, with enhanced expression, start in the mid-age. And he also found that 28 transcription factor target sites significantly overlap with the aging markers and identified using the single-cell RNA-seq, including STAT3, the regulator of inflammation at both mid-age and old. And among them, 16 of them are enriched in motif accessibility by single-cell ATAC, suggesting a high agreement between the 2 assays. And these results show that HSC aging signatures start to emerge at mid-age, not just at a much later age. In addition, we assess how the process of aging impact epigenetic and transcriptional heterogeneity using the Sheng entropy. And even by the mid-age, the HSCs start to display higher heterogeneity than young mice. And this is also true for other progenitor cells. And these results suggest that epigenetic heterogeneity can serve as a biomarker for HSC aging. Similarly, by mining the public data sets, we assess how the TET2 loss impact heterogeneity in HSCs from young, healthy mice using the published data. And indeed, the TET2 loss induced cellular heterogeneity at both the epigenome and also the transcriptome levels. And recently, Marco, the postdoc from the James Lab, very talented postdoc, really helped us to establish this platform to trace the clonal diversification of the aged HSPC in culture, and then applied cell tag indexing of long-term genetic labeling with heritable, virally delivered barcodes to the hematopoietic stem progenitor cells, and then applied single-cell RNA-seq to tag cells after 16 days of ex vivo expansion. And this is with the goal to parallel capture the HSPC clonal history and the cell identity. And beyond the regular serial analysis, Xiaowen performed the cell tag error correction, genomic regions filtering, and cell filtering, and JCAD analysis to detect this clonal ID. And here we hypothesize that cells in the same clone from the old mice are more likely to display higher overall clonal transcriptomic diversifications than young mice. As shown in the y-axis of the box plot here, we use the pairwise correlation coefficient of single-cell transcriptome from the same clone in purple or different clone in green to measure the clonal diversification. So the lower the correlation, then the higher the clonal diversification. And HSPCs from the same clone in purple shows a higher transcriptome similarity than HSC from different clones here, and confirming the robustness of this clonal identification. Notably, as shown here in the purple box of intraclonal correlation, the old HSCs have a higher clonal diversification than the younger HSCs, and suggesting a greater rate of generation of heterogeneity from a single long-term HSC clones. And shortened HSCs and multipotent progenitors from old mice also behave similarly. And these genes are not random, but are enriched in pathways that related to the HSC biology. For example, like this HSC-PC fitness, including the ribosome pathways and also those pathways that are related to the response to inflammation, such as JAK-STAT signaling pathway and the cytokine-cytokine receptor interactions. In addition, here, there's the hematopoietic cell lineage, ML, and pathway in cancer. These are also enriched here. So in summary, here we find that HSPC, epigenome, and transcriptional heterogeneity increase with age. And in the collaboration with James and also Eric, we have shown that established this platform to parallel capture the clone history and also cell identity. And these enable us to also find that there's a faster generation of heterogeneity from a single clone of old mice. And then these genes are not random, but also enriched in the pathways related to the hematopoietic stem and progenitor fitness. And now we are working on established systems to unifying the guiding principles to study the age-related disease and beyond. And hopefully this is not just benefit the age-related clonal hematopoiesis, but also many other diseases related to like heterogeneity. So with that, I would like to also share with you my outlook of like data science in the epigenetic heterogeneity, cancer evolution, and aging. And one of the major topic in my lab now is to looking at aging and the leukemogenesis, and particularly what's the impact of aging on the cancer initiation through the leukemogenesis. And here we are looking at the epigenetic heterogeneity and also in vivo clonal tracing of leukemogenesis through the OncoAging Consortium UO1. And we are also just recently funded by the NIA R56 to looking at the disrupted gene regulations in the clonal hematopoiesis. And along that line, we are also developing algorithms and also database to help us further understand better the epigenetic heterogeneity, especially leveraging the long-read platforms to looking at the haplotype-aware allele-specific methylation and gene regulatory network as part of the Morphic consortium to understand all human transcription factors, what's exactly their like functions when you like remove them. And then lastly, because of the importance of looking into the ecology when you study the evolution, now we are also looking into the aged ecosystem and its modulation, and specifically as part of the cellular senescence network consortiums. Now we are looking at the senescent cell and immune microenvironment through the NIA U54 grant, and specifically looking at the aged tissues and looking at how we can learn from the spatial multiome data and combine with deep learning to better have an understanding of the aged ecosystem, including the immune cell and senescence crosstalk. And then we are also just submitted a recent supplement to look into senolytics and aged hematopoietic systems. So coming back to the long-read omics, I believe this is a new way to monitor cancer evolution and heterogeneity. So long-read sequencing has been named as the method of year in 2022 by Nature Method. And right now there's like mainly two platforms that has been really popular, including Oxford Nanopore that enables 10 to 100 kb read-length sequencing, and PacBio that can cover about like 1 to 2 kbs of read-length. And by combining these multiple long-read platforms, now we are able to get the complete genome thanks to the T2D telomere to telomere consortiums. The first complete genome reference generated that filling the gaps, including 8% of the genome that was previously unmappable because of short-read platforms. And these kind of long-read platforms can enable us to parallel mapping the genetic and epigenetic heterogeneity, because from the same single molecule, you are able to measure the base and also base modifications. And this can be covered by those regions that are previously unresolved regions, such as telomere and centromere, that are of high interest for cancer and aging research. And just last month, a draft human pan-genome reference just released and just really like remind of the importance of like there are many regions in the genome that hasn't been explored because of the previous technology limitations, but now with the long read it can do much more. So my lab has been looking into also the single like the long read platform, especially Oxford Nanopore platforms, to understand the like single base, single molecule long range phase demethylation patterns. And right now we have Yang Liu, a research scientist in the lab, previously postdoc, have developed a next flow pipeline, leveraging the meta-learning algorithms to report the consensus DNA methylation status that are cover the entire epigenome together with the complete genome. And by combining the genetic and epigenetic information, now we're able to, like using the pipeline nanometer to call the haplotype aware allele-specific methylation at all the regions of your favorite that are previous covered, but also telomere centromeres that are not covered by the HG19 or 38. And I would anticipate there's a lot more like low-hanging fruit findings from those regions that just haven't been tapped at all in the past. And another ongoing effort in my lab is to study the disrupted transcription regulation of aging and clonal hematopoiesis. And here, we're particularly interested in how does the CH mutations such as TET2, DNMT3A, and the different age from young, mice, mid-age, and old, how does this aging and genetic cooperation together to disrupt transcription regulations by combining, looking at these intrinsic and extrinsic factors, the inflammatory microenvironment. And specifically, we're applying the 3D epigenome technology, including single cell epigenome, DNA methylome, and CDCF binding using the cat-and-rum, and the single cell transcriptome, plus the chromatin interaction to give us a comprehensive view of the disrupted transcription landscape. Using both the published software from other labs and our own labs that developed for diamethylone analysis and chromatin interaction for high-C and ChIP data analysis. And the goal here is to looking for those driver events, not only in the later stage, but also at mid-age to find like a drug target that can potentially mitigate clonal hematopoiesis and leukemogenesis. And this is a work that in collaboration with my JAX colleagues and also UConn collaborators. And last part is the aged ecosystem, because like nothing in evolution makes sense if unless it's through the lens of the ecology, right? So now as part of the NIH Common Fund Cellular Senescence Network Consortium with my JAX colleagues and UConn collaborators, now we are looking into the age ecosystem and cellular senescence, and that's mainly enabled by the spatial multi-omics. And specifically, we are looking at multiple modalities, including transcriptome by Visium and Xenium, and the proteome using codex and image mass spec, and the lipidome and the metabolome, again, through the image mass spec. And the goal here is to, like, from the perspective of data science, we are integrating the image with omics data through deep learning. Some of the models that we use, such as the vision transformers, are same categories of models that you are more familiar with, such as like ChatGPT. So here, we are combining the deep learning models and using the deep learning model to combine the image and omics data. And through the model interpretation, the goal here is to understand the histological context and tissue morphologies, how that influence the spatial multi-omics landscapes. And eventually, the goal is to understand the spatially heterogeneous age ecosystems and the secretomes that come with senescent cells, and also the senescent immune cells crosstalks that shape the landscape of the aged tissue and eventually drives all kinds of the age-related disease. But this project for now is just healthy aging. So with that, I would like to thank everyone in my lab and also our collaborators across the nation, especially James and Eric from this campus, and also like many other like collaborators within JAX and across the nation. And thank you also for all the fundings from NIH and the internal fundings from JAX Cancer Center, and thank you for your attention.
That works. So I'll start off with a question, but once I ask this, please do wait for the microphone, because the people on Zoom won't hear it if you don't talk into the microphone. So first off, fantastic talk, really amazing stuff. So this is something that actually relates to our own collaboration, but it's always sort of puzzled me. I totally get why aging would be associated with more epigenetic heterogeneity. A lot of reasons for that, but how is it so specific to particular genes? Is that through specific transcription factors like TET2 type thing that are affecting specific genes? So what's your best guess on that? I know we don't have anything hard, but what's your best guess on that?
Yeah, I think it's at least at one level is related to how the chromatin was folding and how it's made it accessible for all kinds of factors to binding, and similarly the other aging related factors to interfere with them. So depending on how open or closed the chromatin is well also related to how it's changed the demethylation landscapes. So that's I think it's one possibilities. And there's maybe other factors just in general those related maybe to the evolution over time, some of the factors that are landscape are more important. You have to really, the cells are developing all kinds of mechanisms to protect them, to ensure that this is not disrupted during aging. So I think that's many factors together contribute to that. Yeah.
Great talk. So I have a couple of questions. The first one is, so both the mutations and the drug treatments that affect heterogeneity also affect the global levels of the methylation. So how do you separate those two factors? Like the heterogeneity could be a passenger for both the phenotypes of disease or for how the treatment works, but it's actually the total level of methylation, even at specific loci, that matters. And the second one was, so it's really cool about the long read sequencing. How does the heterogeneity you get from the older stuff with the short read correlate with when you calculate it across much longer distances?
Yeah, both are great questions. So the first question is about how you distinguish the impact of somatic mutations and the therapies on the epigenetic heterogeneity, right?
I mean, heterogeneity versus just the global levels of the methylation, because you increase heterogeneity, but also if you have a TET2 mutant, you're going to have more methylation throughout, right? Am I right about that? Or if you have the drug that blocks DNMT3, globally, you're going to have less methylation. So how do you separate the effect of just reducing methylation versus changing just the heterogeneity?
Yeah, that's a great question. So we also in our work in published on cancer discovery earlier, that we looking into those regions that don't have the hypermethylation. So based on the cutoff, if there's a region that have a differential methylation percentage less than 25%, that's the general criteria to define differential methylation regions. So we still see there are regions, even without a hypermethylation, you see an increase in the epigenetic heterogeneity. So that you still can see that separated by different genotypes. So I think there are changes that are beyond just changing the hypermethylation, but you also have, if you take another look, the demethylation changes enabled by the somatic mutations in TET2, for example, cause hypermethylation. They contribute to the epigenetic plasticity, right? They create additional status. So this inevitably, I think we need to take into account as the epigenetic heterogeneity to interpret the functional impact of epigenetic heterogeneity. So whether we should really separate them well, I think that's will be the next step. The first step, what does it do? And that's what we are working on now on the U01 grant. And the second question you have is about long read and whether it's aligned with what we find in the short reads. And that's definitely a very interesting question. I think the field is moving towards that, and they still need probably more time to get adapted to that, because one of the barriers, how do you utilize the long read sequencing data? It's different data type already. We deal with the ion current generated from the long read platform. And the good thing is that you are able to predict the base and the base modification at the same time, right? But how can the cancer community more adapted to this new platform? I think that will take time. And one of the effort we do is to develop the pipeline that are not only good for computational biologists, but also just non-computational biologists, that they can use the cloud computing platform like Rapid OS to just click on the different buttons and being able to fire the demethylation analysis for nanopore data. So I think this field need more time to adapt to that. But you already see that TCGA platforms with the microarray and demethylation by cell-based sequencing, it enables a pan-genome research, and tells us how much important is the epigenetic regulators in cancer research, right, in the past. And I would envision there's another wave that enabled by the newer tech platforms. Long read is one, another is spatial omics. There's many emerging large-scale consortiums that are people get together and looking at the cancer in a new way. Yeah.
Sheng, great talk. So good to see you here. Coming back to this idea of epigenetic heterogeneity and environment, what is the impact of the presence of leukemia on the epigenetic heterogeneity of normal cells? Is that a situation that resembles aging in which epigenetic heterogeneity increases as these clones attempt to find a solution around a difficult problem of where to be or how to interact with leukemia? And then, as a related follow-up to that, when we're using drugs like azacitidine, which of course, as a monotherapy, your data show is not all that great, are we in essence using a blunt instrument that likewise heterogeneity or even just simple epigenetic states that they need in order to retain their fitness in competition with leukemia?
So last part, can you say it again? Synchronize them into the same status. And what's the question?
Right. So the question is, what is the orthogonal effect on the normal cells in that situation?
That's a great question. So coming back to the first question, what's the impact of the normal HSC, epigenetic heterogeneity increase with aging? What does it do to the leukemia genesis, right? I think it will be multiple layers. So first of all, they provide a higher heterogeneity foundation for those mutant cells to generate more transcription program they can sample, right? The hypothesis here is that the aging and the mutations such as tattoo, they cooperate together to increase the heterogeneity. So if you just have the tattoo without the aging impact, I would anticipate that this heterogeneity level will be lower than the tattoo and the aged mice. And we are there's an ongoing effort in the part of a study to looking at dissecting the impact of aging and the somatic mutation, right? So I think these two together will further enhance epigenetic heterogeneity and thus increase their a chance of adapting to the aged context. So that's one layer of the impact from the intrinsic level. And the second is the competition between the normal cell and tumor cells, right? And what we hypothesize in our collaborative project is that if there's increased heterogeneity in the tattoo and the aged mice context, because of the much higher heterogeneity compared to the wild type, because they don't have somatic mutations, there will be higher chance for them to win the game eventually, together with the impact bring by the DMS, the tattoo methylation, tattoo mutations change the 5MC landscape.
Yeah, and definitely makes sense. I just want to clarify. Do you suspect then that the wild type or at least non-leukemic cells in competition with the leukemic clone would experience even greater epigenetic heterogeneity than they might if it was simply an old individual without any leukemia? In other words, does leukemia itself prompt changes in epigenetic heterogeneity in normal cells that are competing with it?
That's an interesting question, and you can also think about the cell-to-cell interactions. How do they communicate with each other? So that part we still need more data to looking at. Anticipate once we start to dive in more on that, I think there will be a lot of questions can be answered in this angle, more specific biological questions. And I think the experimental design currently we have should be able to answer this kind of question, but we haven't explored that yet. Soon, no.
Back here and then over to the seconds.
So in the context of aging, do you think microbiome can also have effect on the epigenetic heterogeneity? Because I guess there are some microbes that produce ultimately SAMs, and that can basically be used by the methyltransferases, right?
Yeah, I would anticipate that's the case, but exactly how they impact. I know that these are the questions that has been answered by some of my colleagues at JAX, and not directly related to age-related clone hematopoiesis, but on the those cellular heterogeneity in the skin or others. So I think it's definitely plausible questions to ask. And more direct evidence was seen in the colorectal cancer, right? When you see that there's a tattoo mutations, and then you change the microbiome, and you see what changes. I remember there are papers published on that in that interaction. So it's definitely possible questions, but we just don't know yet. Yeah.
Just one quick question on. So are there efforts now towards more bisulfite-free sequencing technologies like TAPS or something like that, tate-associated methylation? Because bisulfite sequencing is just too much DNA damage, right?
So depend on the context. So say like when we look at ML specimen, usually they have more DNA to work with compared to liquid biopsy in the plasma, much less DNA methylation. So there's a lot of research ongoing in what's the better way to analyze that. Even for bisulfite sequencing, there's ways that you can reduce the damage to DNA. And that's I understand this is an ongoing effort. People really want to decrease the input amount for the bisulfite sequencing. Yeah.
So in real life, we have aging, but aging is not that we just age over time, right? So during that time, you go through multiple infections and recovery. So is it just aging, or it's just exposure to multiple, if you want to call danger signals of anything like what happened in the epigenetic landscape? For instance, if you take a young mouse and you infect first with the flu, recover, then you give a Pseudomonas, recover. What happened in the HSC? What happened in the cells in general? Do you have a change? Because say in real life, it's not like, oh, we are good for, since we are 5 to we are 80. You go through multiple rounds of colds, flu, COVID, anything. So how that can actually influence the HSC and other cells?
Yeah, that's definitely a valid question. Like, what's your lifestyle and infection history shape your epigenome, right? And that's something that I think has been really studied a lot in the population genetics in terms of like measuring the demethylation and associated with the lifestyle, like whether you smoke. There are, like, I just recently come to knowledge, like these, you can find a set of demethylation markers that predict whether you have smoked or not. So that just makes me believe that there's a lot of information already encoded in the methylome and in epigenome in general that you can eventually shaping how our like epigenetic heterogeneity landscape comes when it comes to the like a cancer initiation. Yeah, but that's definitely a valid question. Like maybe one day we'll all have to categorize the lifestyles associated with DNA methylation markers that help us to better understand the functional impact of epigenetic heterogeneity. And that's something that also has been in my mind, like I would like to incorporate and enhance the measurement of what we're developing previously in the lab. Yeah.
All right. Well, thanks to Sheng for a fantastic presentation.
Thank you, everyone.